Hello everyone, and welcome to another episode of Monica's Worm Corner. On today's segment, we're going to be talking about RNA interference, which is also known as RNAi. So RNAi can be used by many organisms to regulate gene expression. It can also be used by scientists to figure out the function of a gene, and also to identify molecular therapeutics that could prevent disease progression. And so I have drawn out the central dogma on the left-hand side of the board. And so the step that RNAi interferes with is this RNA step. And so what it does is it silences RNA so that the protein can't be produced. And so in our lab, how we conduct RNAi experiments is we feed double-stranded RNA to our worms. And so once that double-stranded RNA enters the cytoplasm, it gets cleaved by dicer. And so what dicer will do is it'll cut this double-stranded RNA into these small fragments that are about 20 base pairs. And so these small fragments are known as small interfering RNAs, and they're called siRNAs for short. And so there's also another small RNA, and that's microRNA. And so what microRNAs are is that they're basically RNAs that are folded and processed in the nucleus before they're exported into the cytoplasm. And so with siRNAs, what happens is that it'll bind to an argonaute protein, and what binds is this guide strand. So this combination of a guide strand, the argonaute protein, and other proteins forms the RNA-induced silencing complex, which is known as RISC for short. So RISC, which is being led by a guide strand, will then complementary base pair onto an mRNA target sequence. And with siRNAs, this complementary base pairing is very precise. And so once it binds to that target sequence, it'll cut and degrade that target sequence, and then the protein cannot be produced. And so microRNAs can also guide RISC onto target mRNAs. And so with microRNAs, only a segment known as the seed will complementary base pair onto the target sequence. And with microRNAs, the complementary base pairing is not very precise, or is not as precise as siRNAs. But this imprecision allows for microRNAs to target like hundreds of endogenous mRNA sequences. And so that was a short introduction on RNAi. I hope you all enjoyed. So thanks for watching.